Thanks, Mike. We're here with a special, what I would call a spring break edition of Pitch Club in surgery and cardiac surgery. Part of our team is gone. I'm actually coming to you from the state of California as opposed to my home state of Colorado. But welcome, everyone. We're glad you're here tonight. And we have three great companies lined up. So without further ado, I'm going to introduce our first presenter. And this is Mr. Almog Ale-Raz, and he is the co-founder and CEO of a company called Corneat. Welcome.
Thank you. Good evening. My name is Almog Ale-Raz, and I'm the CEO and VP R&D of Corneat Vision. Today, I will present you our disruptive biomimetic implants technology. It is poised to significantly impact implants and implantables and bring about novel solutions that address unmet needs across multiple therapeutic fields. Our core technology was discovered serendipitously 6 years ago by Dr. Gilad Litvin, who's my mountain biking teammate. Gilad, who's an ophthalmologist, was working on an artificial cornea and was researching ways to permanently integrate artificial optics with resident ocular tissue. The main issue with implants and implantables is, of course, the foreign body response they trigger. Our immune system will work to either degrade and eventually absorb foreign or implanted material, or alternatively encapsulate it with a granuloma. Devices that aim to integrate with tissue, for soft tissue reinforcement, for example, eventually get absorbed. No one is set to find a way to seamlessly and permanently integrate synthetic material and implants with surrounding tissue. Well, until now. I'm honored to present you today the EverMatrix. The EverMatrix, which is seen here, is the first ever non-degradable material that mimics the extracellular matrix, which is essentially the collagen skeleton of the tissue. When implanted, its structure and polymer composition enables it to practically go under the radar of the immune system and stimulate cellular proliferation that leads to full integration. The material can be fabricated to thicknesses from dozens of microns and has extremely high tensile strength. The fact it remains in the tissue makes the integrated tissue even stronger than the original, kind of that like metal mesh within a concrete wall. In January 2020, we hit a major milestone proving EverMatrix's ability to biomechanically integrate substance and tissue. Jamal, a terminally blind 78-year-old man who didn't see for a decade, in January 2020, we hit a major milestone proving EverMatrix's ability to biomechanically integrate substance and tissue. Jamal, a terminally blind 78-year-old man who didn't see for a decade, regained his sight following a 1-hour surgery. This truly emotional moment, which was captured on video, was broadcasted on TV news and on social media all over the world. Jamal is now 15 months post-op, and his vision is stable since day 2, with no complaints on pain or discomfort. When our clinical advisors were first exposed to our animal studies, they immediately indicated they'd love to get a piece of this material to deal with other indications. We've pivoted since, and today we have 4 products under development, proving that EverMatrix can be deployed to support several use cases. While we started with biomechanical integration of implants and tissue, EverMatrix can also enable permanent soft tissue reinforcement. We can use it as a membrane for guided tissue and bone regeneration, and also for concealment of irritating implants and sensors. Applications are spanning ophthalmology, periodontology, orthopedics, general and plastic surgery, gynecology, and more. What makes EverMatrix work is its similarity to the ECM in terms of fiber diameter and porosity. As you can see in these scanning electron microscope images, it looks the same. We scanned EverMatrix next to a strain of hair so you can understand the dimensions. The fibers are 1 to 2 microns in diameter. But it's the porosity that triggers fibroblast migration. Pores in the range of several microns enable cells to exert forces on the ECM, sending mechanical signals that propagate in the matrix. The mechanoreceptors on the fibroblast membranes react to these forces, triggering biological processes that lead them to protrude along the matrix fibers and eventually latch onto them. EverMatrix pores are smaller in an order of magnitude than any non-degradable materials tried before for tissue integration, PTFE, for example. EverMatrix fibers are non-degradable as they are made from polyurethanes. We use a chemical engineering process called electrospinning to make them. This is a well-established method that is used today to produce collagen and other absorbable scaffolds. but it's the permanency of Evermetrics that makes a difference. Evermetrics' disruptive tissue integration capabilities were proven in over 12 different studies, in vitro, in vivo, and now also in humans. Here in the center, you can see histological evidence from an in vivo study demonstrating full fibroblast colonization and abundant collagen deposition within our matrix. There's no capsule formation, and even capillaries coursing their way through the material, rendering it an integral part of the tissue. Our Everpatch product, which is a permanent tissue reinforcement matrix for ophthalmic surgeries, here on the right, was used to cover and conceal a glaucoma device laid on the sclera. As you can see in our first patient, who was operated in both eyes, the eye with the Everpatch is quiet, while the other with the donor tissue is congested and inflamed. Besides the huge aesthetic differences, our key advantage here is, of course, permanency. We currently work to clinically validate our first two products, teaming up with leading clinical sites in the US, Europe, and Asia. We expect our first FDA regulatory approval within a year. We have a very strong broad IP. Our patent and IP strategy were recently validated by Wilson Sonsini, who also recommended drafting additional divisional patents for some solutions. So far, we've raised $12.5 million. Additional $4 million were invested last year from Biomed VC with focus on the Chinese market. We were recently awarded a $2.9 million grant from the European Innovation Council to support scale manufacturing and market introduction of our artificial cornea. We are currently seeking $15 million in the form of a SAFE that will bridge the company to round B, planned toward the market launch of our artificial cornea. This will enable maturing the company toward an IPO or M&A 18 months later. We already have established contacts with some potential buyers who are following our progress. We have a very devoted, skilled, and efficient team, which includes engineers, clinical, regulatory, and business experts. From a scientific perspective, we are supported by the world's top physicians. These include current and past presidents of leading medical societies. We are looking for leaders and KOLs to support us with R&D and clinical work, as well as with investment, enabling us a soft landing in the United States. We invite you all to join us on our unique, exciting, and life-changing journey. Thank you.
Almog, thank you. As I mentioned, please put your questions for Almog and Corneat in the Q&A function, which is next to the chat. I'm going to start us off here. This is really exciting technology. I just look at it and I think it likely has so many applications. And I know you've started with ophthalmology, but given the permanence of your Evermatrix, I'm guessing this could be used even in high stress areas such as tendon repair.
Yeah, I mean, every physician that sees the results, it's for ophthalmologists, it's an eye opener. For periodontologists, it's a jaw dropper. And we've been speaking to gynecologists and orthopedics. Everyone has use cases that are brought up immediately when you see the histological results. Of course, eventually, if this integrates with the tissue, giving it strength and flexibility, this can really be a game changer also for these use cases.
That's super exciting. And tell me, I know you have implants that are in humans at this point. How long have you followed those patients out? Talk a little bit about your clinical outcomes so far.
Yes, so for the Everpatch, that little piece of that material for ophthalmic surgeries, we have 26 patients now. The longest follow-up is 16th month. This study is going to be closed soon, and we'll be following up all patients for another 6 months. We are also running a clinical trial for artificial cornea with 8 patients implanted already. Most regained sight. There are some amazing stories out there. We have patients in Canada, in France, and here in Israel. We'll be closing this first in-human study, just doing a couple more implantations and start a pivotal study with another 30. This will put us on a path to a regulatory approval within a year and a half to 2 years.
That's fantastic. And I think a game changer for those patients, I'm sure. We have a couple of questions from our audience here. One, don't polyurethanes undergo metal ion-induced oxidative degradation, over time? In parentheses, over years, really is what the question is.
Yeah, so polyurethanes are very stable. They're used today, I think, to cover pacemaker leads, for example. It's a very well-known material, and it doesn't degrade over time. We are using a carbonated polyurethanes, which are even stronger than just simple polyurethanes. And so far, we haven't seen any degradation in none of our studies and preclinical work.
That's exciting. And again, how far out have you followed that up to this point?
So the material, I mean, this polymer doesn't go anywhere.
Okay.
I mean, we've done exhaustive extractions and long stability testing. In the ophthalmology space, we need to do hydraulic stability tests that simulates about 5 years in the human eye, and no degradation whatsoever.
Very good. All right. What's the 12 million that you're raising currently going to be used for?
So mainly it's going to support the existing clinical trials of the first two products, and also the initiation of clinical trial with the gingival reinforcement patch, and with the eShunt, which is a glaucoma drainage device that integrates with the eye wall. We're also working on scaled manufacturing, as I mentioned, and we will need to do some market access work. So working on reimbursement and distribution. We actually signed up our first 3 distributors last quarter, and we hope to enter the market with enough distributors on the ground to support our numbers.
Very cool. And another question from our audience. So with the artificial corneas, if your device is not used to kind of hold the artificial cornea in place, what happens? So what's the benefit of utilizing that EverMatrix in that procedure as opposed to not, and how long does it last?
So the way the Corneat Capro, the keratoprosthesis work, is that we have a lens which is very wide. It actually resembles the original cornea, and it's much better in terms of optical quality than anything that was ever done in this space before. This lens is surrounded by a skirt that is implanted under the conjunctiva. So it does hold the lens in place. We have a posterior undercut where we fit the corneal stump of the host cornea, but this is only good for short term. For the long term, we rely on biology for the integration.
And in cases if your product wasn't used, what happens to those corneas? What are the concerns that we would worry about?
In cases where the Capro wouldn't be used, well, for corneally blind patients, the only available solution today is keratoplasty, donor tissue. I don't know if you know, but about 20 to 30% of these surgeries are repeated surgeries, and there are about 20% of the indications that are not suitable for clinical reasons for the surgery. So while in some places, like in China, there are 5 million corneally blind patients and only 5,000 transplantations, even in the US, there are about 100,000 people on the sidelines that are not in line for the surgery. We can provide these people with their vision and independence.
That's fantastic. Well, Almog, I appreciate you being here tonight. It was great to learn about Corneat and what you're doing. And just to know that not only is this being used in the ophthalmology space, but you guys are expanding well beyond that. So super exciting. And thank you again for being here.
Thank you for inviting me.
We have our next speaker coming up to the stage, and I'm excited to introduce Dr. Greg Grover. Greg, thank you for being here tonight. He is the co-founder and CEO of Kairos Technologies.
All right, let's get this fired up. Hi, everybody. My name is Dr. Greg Grover. I'm the CEO, co-inventor, and co-founder of Kairos Technologies. The core of Kairos' technology platform was some cross-linking chemistry I invented as a PhD student in organic chemistry at UCLA. I then took this chemistry down to UCSD as the American Heart Association postdoctoral fellow, and there I worked with bioengineers and cardiothoracic surgeons to understand problems more deeply, how we could use this platform technology to address unmet needs in surgery. And the first focus that we wanted to address with our technology were post-surgical adhesions. We view these as the holy grail of post-surgical complications because this is what surgeons have told us. 5 out of the 10 most common surgical procedures are significantly impacted by these post-surgical procedures or post-surgical adhesions as shown on this slide. Now, there are two approaches to deal with these adhesions. The first is a solid film or a solid membrane, like Sepra film, for example. You can think of this like saran wrap. These films suffers from issue of dislodgement, rapid degradation, and they're also difficult to prepare and use. Imagine saran wrap covered in water while wearing gloves trying to wrap it around an organ. The second approach is to use a viscous hydrogel solution. So the concept here is you separate these two tissues so that they can't interact with each other. These gels suffer from high swelling, rapid degradation, and there's also issues with preparation at the surgery site and their use. And so the key here is that none of these approaches have been effective. And so reoperation is required to remove these adhesions. And so we first chose to focus on the heart. And here we invented Tissue Shield, a product that's designed to protect organs and tissues. And our goal here is to protect these patients and save hospitals money. The illustration on the left shows our figure being used on a heart. And here we're going to show our prototype being used in a surgical setting. This is a partial sternotomy in our pig study. You can see that the prototype easily fits in the surgeon's hand, and it's easily sprayed over the surface of the heart, rapidly gelling and creating this protective coating. It's important to note that cardiac adhesion removal triples the rate of injury and death on reoperation for these cardiac patients. So we really wanted to focus on this area because there are not many solutions to address this need. The core of our technology is this novel 3-component hydrogel platform. The first component is this resin here shown in pink, this activated aldehyde that interacts with our hardener shown in red. This is what cross-links and forms the hydrogel. What's unique to our technology is we have an added tissue binder derived from the muscle binding protein domains, DOPA. And so this reacts with tissue surfaces in a liquid state, forming a protective coating that when cures, prevents cell and proteins from binding. We've developed this dual chamber, dual syringe, ready to use solution as shown here at the bottom. When we were doing feedback analysis from surgeons, it said they wanted something easy to use that would require no assembly or preparation. We have great preclinical and safety and efficacy data. Here I'm showing figures from our Nature Communications paper from last year. The graph on the top left shows how we prevent cellular attachment to the formed gel. The graph on the top right shows how we're cytocompatible and not toxic to cells. The graph on the bottom left is from our rat study, where we show that we prevent adhesions in our cardiac rat model. And then the figure on the right is a histology slide, where you can see that the material after H&E staining is still bound to the surface of the heart. Our technology is supported by 3 patents with exclusive commercial licenses from UCLA and UCSD and 4 peer-reviewed publications. We're also currently developing our own IP and growing our portfolio as well. So we have a proven prototype and a clear timeline. We were founded in mid-2020 based on the results of these rat and pig studies that we published with our prototype. And I mentioned 3 patents issued. We're raising a seed round right now of $2 million, where the key takeaways are to scale up our manufacturing for this prototype, this dual chamber prototype here, as well as performing a piglet study, which we just received a score of 25 on our NIH SBIR phase 1 grant. We'll also be doing FDA submissions. We have fantastic collaborators and partners, including leaders in this space, including Credence Med Systems, Lyophilization Technologies, and West Pharmaceuticals. Our goal is to get into clinical trials as quickly as possible, with the first focus is a first-in-human study of 30 patients. Our initial area that we're focusing on are children with congenital heart defects. This is because this type of patient has a staged operation where they require that second operation anyway in 3 to 4 months. Therefore, we can do a 1-to-1 study there. We also are looking at moving on to a pivotal study, but our goal is an acquisition by a strategic partner such as those shown here, J&J, Baxter, and Ethicon, for example. There are potential exits at each stage here after our clinical studies. We have an experienced and successful team. I personally have developed other hydrogel products, most recently as CTO at Contraline, where I developed another injectable hydrogel system to occlude the vas deferens as a male contraceptive device. We have surgeons, former entrepreneurs, as well as those that have worked in spaces such as Boston Scientific, FDA, and research hospitals. We're currently raising a seed round right now, as I mentioned earlier, of $2 million. We have over $1 million committed. And as I mentioned, an SBIR grant that we're looking to receive funding on with a score of 25. Our goal here is to change the paradigm of post-surgical adhesions by giving a technology that can prevent them in the cardiac use case, starting with congenital heart defect patients. However, we know from talking to surgeons that we can also use this product on other indications such as orthopedics, tendon surgeries, and OB-GYNs such as hysterectomies and obstetrics or C-sections. So we're really excited to present our technology to you and would love to talk to you more about what we're doing at Kairos. Thank you so much.
Greg, this seems like a game changer. Super exciting. As you mentioned, preventing surgical adhesions would be amazing for a lot of patients. Better outcomes at a lower cost would be my guess. I'm going to start with a couple of questions. You mentioned that your market entry point is with pediatric cardiac surgery. Woohoo! As a pediatrician, I love to hear that. And tell me why you're starting there. I'd like to learn more about that.
So in examining the landscape here and looking at other products that have been attempted in the abdominal and gynecologic space, we saw an opportunity with cardiac because number one, these surgeries are very expensive and life-saving. And for the congenital heart defect patients, specifically single ventricles, they have a staged operation set where they have a first operation followed by a second operation at 3 to 4 months. This will allow us to utilize a novel technique of non-invasive imaging to first assess the adhesions with imaging and then perform the reoperation to assess the product's performance versus no treatment groups in a small cohort.
You know what? That makes a lot of sense. A lot of sense. I know exactly what you're talking about. All right. So tell me a little bit more as well about the key differentiators of the Kairos product compared to other products that are on the market.
Right. So the big players in this space are really two films, Seprafilm and Interseed. And these are, like I said, things like saran wrap that you try to wrap around an organ or, pack into place in tissues. They're very difficult and they don't work very well and degrade rapidly. There's also viscous solutions that just don't do the job and can't translate to the cardiac space anyway because it drains. The secret sauce for us is our cross-linking chemistry is high efficiency, resulting in an integral network, which means we last 6 to 8 weeks in vivo, allowing the body time to heal. We also have that added tissue binder that in the liquid state coats the tissue when it's applied and keeps it in place so it doesn't dislodge like other sprayable products like Co-Seal and Duraseal, for example, when they're sprayed over large volumes. And the real key here is that we don't swell versus these other hydrogel products that swell 400 to 600%, which in a previous pediatric cardiac study caused tamponade.
Oh my gosh. Okay. So potentially bad outcomes with the other products as well.
Correct.
One of the things
AEs.
one of the things that you mentioned actually leads into the first question from our audience. Are there cellular issues with the surface where the hydrogel is applied? What kind of data over time do you have on potentially this type of issue?
Yeah, so I've published implanting this in the sub-Q space, and we have non-published data in the IM space, but we have mice, rat, and pig data, oh my gosh, in the cardiac space. And what we've seen in all of these is that it's biocompatible, biodegradable, with no chronic inflammation response, and that we see a very typical response you would see from these benign materials, so to speak.
All right. And another question from our audience. How does your product affect wound healing, if at all?
Yeah, we did not see an impact. So that's a metric that we'll be measuring in that piglet study that I talked about, as well as a number of other key facets. Come by the table, and I'd love to tell you more about this great piglet study that we designed with our medical advisors who are chiefs of cardiac surgery across the country. But no impact.
All right. So another question around, would the product get immediately suctioned and or drained by chest tubes or drains, depending on where the surgery is, in a variety of different places?
Love this question. Great question. So our product gels in less than 3 seconds. So that's key. We tuned the gelation mechanism. As you saw in that video, you could see it gelling rapidly. You could see a little bit of the gel at the top of the rib cage or the entry there. And so we gel immediately upon contact with the tissue, curing and forming that barrier that protects it. And then that's really key. And we don't dislodge or fluff off like other products.
And the reality is that product stays in place until the body's natural healing is that 6 to 8 week window, correct? Where the body's natural healing is taking place at that point.
So I'm not a surgeon, right? My partner is, though. And then from talking with other surgeons, we understand the big window is that 4 to 6 week time period, which is why we invented a product that lasts 6 to 8 weeks, giving the tissue time to heal, the body time to reach homeostasis. However, it is important to note, this was a feature that Dr. Madani, our co-inventor, was so excited about, is he wanted to be able to peel it away if he had to. and you can mechanically remove it if you need to, as we show in our Nature paper.
All right, very cool. So one last question, which I think is a great one that I had as well. So beyond cardiac surgery, what other conditions are you looking at? Where else could this be used?
So the two next areas that we've received a lot of interest are in orthopedics, specifically hand tendon surgeries. The surgeons in this space that I talked to, the hand surgeons, think that we can prevent that second surgery where you need to release the tendon. So they're really excited about that, and we're working on developing an animal model and a grant submission there. Another area that I'm very passionate about is we want to go after C-sections and hysterectomies. This is a really important area that affects hundreds of thousands of women in the US alone and can actually prevent them from having additional pregnancies. And the current products are just not effective enough. So in summary, we're going after the CHD cardiac market. This will expand the ability to use it in other cardiac cases because of our non-invasive imaging. So we won't require the reoperation. Come by the table, I'll explain that in more detail. And then we're going to stagger SBIR grants and STTR grants for those other indications. So if you're interested in collaborating, please let me know because it'd be great. Sorry, getting it all out there.
No, you know what? I love it. Greg, thank you so much for being here. I know Dr. Macaluso, who's asked a couple of our great questions tonight. He'll be at your table afterwards because that last one was one from him. So, Greg, thank you again for being here, and thank you for telling us all about Kairos. It's really exciting, and again, potentially a game changer. Excuse me, in the field of surgery, right?
Appreciate the opportunity. Thank you so much.
All right. And I am going to bring us back to our pitches of the evening. So last but not least, I'm going to introduce Greg Aber, and he is the CEO and CTO of Corvion.
All right. Hey, thank you, everyone. So Corvion is developing what we consider is the first safe and reliable, fully implanted LVAD. LVAD, for those who don't know, is left ventricular assist device, basically a small pump that's implanted to assist the heart for patients suffering from severe heart failure. LVADs have been around for decades, but recently, I'd say the last 10 years have really become a standby or a frontline therapy for heart failure patients waiting for transplant and those who are ineligible for transplant. So the current market is dominated by one device and one company, that being the Abbott HeartMate 3, which was acquired through several acquisitions going back to Thoratec. Medtronic was in this space, but recently bowed out. So the Abbott device sold market share, basically 100%. Small pump implanted in the left ventricular apex with an outflow graft that's anastomosed to the aorta. This device requires a lot of external equipment because it has a very high power consumption, about 10 watts. And all this equipment, along with what's called a percutaneous drive line, which is a power cord going through the skin, results in a low quality of life. And on top of that, has a very high rate of infection where the drive line penetrates the skin, leading to high rates of stroke and bleeding caused by lack of pulsatility of this device. So we're approaching this from a different standpoint, that being fully implanted with no power cord through the skin, no external battery packs, everything implanted. This has been kind of the holy grail of this field for 30 years or so. And we feel the big stumbling block has always been the power consumption of the pump. So we focused on developing a very high efficiency pump design, about 5 to 6 times more efficient than the HeartMate 3, around 1.5 watts. So this low power consumption then allows for an implantable power source about the size of a large pacemaker. And because it's not holding much energy, we're able to wirelessly recharge it, similar to an iPhone, in a very convenient way for the patient, with a lightweight device requiring no power cords, no contact with the skin. So why is our pump so efficient, right? I mean, there's a lot of LVADs that have been developed over the past few decades. We focused on a different design than basically anyone has ever attempted, in which the pump is separate from the motor that drives it. And so we're able to have a very novel pump impeller geometry that drastically reduces the torque required to spin the impeller and therefore the power. And then we couple that impeller to a motor with a magnetic coupling. So the motor is completely separate and does not see blood of any kind. So only the pump chamber sees blood. And then lastly, we have to levitate the impeller so that we don't have blood damage. And we're able to do that with small permanent magnets rather than large electromagnets, which are used in the Abbott device. And we're able to maintain a very stable levitation of the impeller with just permanent magnets, no sensors or electromagnetic coils required. This allows for large gaps above and below the impeller for blood to flow in and out of the device without creating high shear. Now, although this may seem like a fairly straightforward design, we have been able to have over 40 key patents granted and issued for the pump and the system design because no one really thought a device could be designed with a separate pump and motor and still be small. And so that's where our key technology lies. So a little bit about our preclinical data. We basically compare everything to the Abbott device. You can see here hemolysis testing on the left. You know, as you lyse red blood cells, you release hemoglobin into plasma, and so the plasma becomes more red. So this is direct comparison of our device to the Abbott device, where we pumped the same volume of blood and from the same animal at the same time. And you can see our plasma stayed much more clear. And also in the analytical data, our free hemoglobin levels were about 70% lower. We then went on to animal trials, which we're currently in the middle of. The pictures in the front or in the middle here are of the pump directly out of our first chronic animal study. No thrombus, no deposits, perfectly clean pump. Our free hemoglobin levels stayed at normal, as if there was no pump in the animal at all, at around 1 to 2 milligrams per deciliter. Power was 1.5 watts and flow was 4 to 6 liters a minute. So pretty remarkable. This really type of data has never been seen for an LVAD. Okay, so we started actually back in 2010. I was actually working at NASA at the time, and where I had developed a blood pump, actually the first blood pump ever implanted in a human previously. And so I began working at home on a new design of an ultra efficient design pump. And that took about 2 years. We received some small family and friends financing along with a small grant from HHS. We then transitioned to animal studies where we did our first bovine studies in the 2019 timeframe. And also about 5 years of product development was required to get the pump refined along with the wireless charging system and the implanted electronics. We knew we had a long road to get the product developed, so we had a large seed round of 11.6 million. We're now collaborating with Texas Heart Institute to finish our animal trials. We've completed all of our bench testing and engineering testing, and the system is working very well. We also have started a small project for a much smaller pump called Nautilus for partial assist, using the same implanted controller and the same charging system. And we recently got a phase 1 grant to start that effort. We're currently in the middle of a $12 million fundraising round. We've raised about 8.6, and we're hoping to complete the remainder of that. So we're looking at first in human around 2024 and begin a PMA trial shortly after that with FDA approval, hopefully around 2026. We've already had some initial discussions with Abiomed and Medtronic. There is some interest there. So I think they're waiting to see how our product performs in humans, and hopefully that'll be positive. So we have a small but highly productive core team of previous entrepreneurs and engineers that have worked in the LVAD field, coupled with some real thought leaders and KOLs in the field of mechanical assist, with Billy Cohn probably being the most well-known, one of the worldwide leaders of this field for several decades. So that's all I had for the presentation. So I'll be happy to take any questions.
Greg, thank you so much. For those patients with severe heart failure, I can imagine, again, the reduction in infection rates and the quality of life issues. You don't have these battery packs that you wear around. Instead, that can be fully implanted. That's super exciting, I think. And I mean, it seems almost unbelievable, right? Because what I heard you say is that what you did here is kind of a simple change in the geometry of the pump allowed it to be much more efficient and allowed the whole device to be fully implantable. Is that correct? Tell me more about that.
Yeah, I wouldn't say it was simple, but it was. It looks simple to other folks because it's similar to pumps you'd see in industry rather than a blood pump you'd plant in a human. But I think the key was always keeping efficiency as the prime focus and never allowing anything else to compromise that. And so in the end, we wound up with a pump design that is similar to an industrial pump you'd see at a water plant or a chemical plant, but is very small and compact and is able to pump blood without creating blood damage.
That's amazing. Congratulations. Super exciting. We have several questions from the audience, so I'm just going to jump right into those because some of them are the same as my questions.
Okay.
Talk about the wireless charging system and how long does a single charge last?
Oh, that's a great question. So the wireless charging system, rechargeable medical devices implanted, it's not new. Neurostimulators have been around for a while, but they all require a coil to be taped to the patient's skin, which is fine if you're doing it once a month, like you would be for a neurostimulator. But for an LVAD, where you're doing it every single day, taping a coil to the patient's skin and then having to peel it off would become very problematic, we think. And so we designed a system that doesn't require skin contact, doesn't require precise placement, can be worn through clothing. So it can be worn on the outside of a shirt or a jacket. And is easily removable. No connectors, no cables, no separate battery packs. Just imagine like a Walkman, from the 80s, where you just wear it on your stomach and you remove it whenever you want. And so the key technology there is highly proprietary, but it involves some physics related to magnetic resonance coupling, which is a way to transfer energy over distance, large distances. And so we adapted that to an implantable medical device so that patients no longer have to have things taped to their skin to recharge the implant.
Well, it sounds ingenious. I love it.
Single charge of the implanted controller will run the pump for 12 hours. So now, patients currently have to plug into the wall at night and go to sleep with controllers and cables. With our device, they'll be able to sleep through the night without having to wear any medical device whatsoever.
That's awesome. All right. So another question from our audience: how long does the pump itself last? And kind of a follow-up to that, is this a destination therapy?
Great.
Meaning, is this for folks that are not candidates for heart transplant?
Yeah. I'm sorry. Could you repeat the first question?
How long does the pump itself last?
Great. Yeah. So basically indefinitely. There's no contact of components inside the pump. Nothing touches each other inside the blood chamber. In the motor chamber, the bearings are made out of hardened ceramic materials, something you'd see like a Swiss watch. And so we think minimum of 20 years, probably closer to 30 years for the life of the device.
Wow.
So basically well beyond the lifespan of a patient that would need one. And then I'm sorry, the second part of that question was?
Well, again, is this a destination therapy?
Yeah, definitely. Yeah. This device, it can be used for bridge to transplant, but those patients usually only have an LVAD for a year or two. They can cope with power cords and batteries easier because they know they're going to be getting rid of it eventually. So this is really geared for patients that are going to live with a heart pump for the rest of their life. 10, 20 years.
All right. So 2 other clinical questions I want to get to here. Is anticoagulation required for patients that have this implanted?
Yes, definitely. I think with any LVAD, you're always going to need anticoagulation. Hopefully with ours, you're going to need less because of the reduced blood damage and the increased efficiency, which minimizes the amount of time that blood is actually in the pump.
Okay. And then another question, is it continuous flow or pulsatile?
Great question. So it is continuous flow, but because of its greatly increased efficiency, we're able to transfer the native pulse of the heart much more efficiently. so that the resulting pulsatility of a patient with an LVAD is about twice with our device versus competitive devices. And on top of that, because the pump's so efficient, we can vary the speed of the motor to track a patient's pulse to add even additional pulsatility. But we really think that's not going to be necessary. We think the native pulsatility will be sufficient for most patients.
All right. So I'm going to say thank you at this point. But for those of you in the audience, I want you to come ask Greg about something else that was on his timeline. And that was the Nautilus partial assist pump, which I think is another product that they are just working on at this point. But I'm going to throw that out as a teaser. Come ask Greg about that after we're all done here. Greg, thank you so much for being here tonight and telling us all about Corvion. Again, for patients that need this, holy cow, a game changer. Amazing. Again, as Mike said at the beginning, we're here every 2 weeks, same time, same channel. And to sign up for the series, go to angelmd.com, because if you miss and you're not able to come on a Tuesday night and join us, if you register for the event, you will automatically get that follow-up communication from us.
And remember, just because it's in a space where you're not used to that specialty or that topic, doesn't mean there aren't amazing things to learn and amazing companies out there. In fact, one thing that Katie and I have learned over the past year and a half of coaching these companies is there are really amazing companies everywhere. And sometimes they surprise you with, like, you never even thought about that as being a potential investment or a potential startup to track after. And yet you learn more from them than you do from the ones you're familiar with. And I just want to throw one last advertisement out there. You know, Pitch Club is really all about spreading knowledge and spreading these the innovation and supporting these companies. So if you like Pitch Club, if you think Pitch Club is cool and some of these companies are neat, well, I'm a total geek for this stuff, so I think they're all neat. But if you think this is a valuable thing, be sure to invite peers and friends. I mean, that's the whole point. It never disappoints. There's always quality companies. There's always something you learn from it. You can always have it on while you're watching March Madness over on, you know, the TV in the room if you want. We're not offended by, like, you watch, you know, playing with your phone while you're watching Pitch Club. But make sure you show up and come by, because these companies love your support, need your support, and it's the way that we're going to get this innovation put into place and useful for society as a whole sooner. Sorry, and I'll leave that as my final advertisement there, Katie. Sorry about that. You can send us out.
Thank you all for being here tonight. Thanks to our 3 presenters. They all did an amazing job. Please join us in the networking right now, and we'll see you in 2 weeks for Pitch Club Innovations in Oncology. Thanks, everybody.